This is our fourth class on molecular basis of inheritance. In our previous class, we have discussed that the DNA replication occurs by semi-conservative mechanism. In this particular class, we will discuss the molecular basis of DNA replication step by step. That means the molecules required for DNA replication and the event sequences of DNA replication. Let's start. In 1955, Kornberg and his colleagues isolated an enzyme that is necessary for DNA replication. Originally it was called Kornberg enzyme, but now is called DNA polymerase 1. That is DNA Pol 1 in short. So what is DNA polymerase? DNA polymerase is an enzyme that catalyzes DNA synthesis. But how? It adds new nucleotide. Suppose this is a ribose sugar, that is pentose. This is the 1' carbon, this is the 2', this is the 3' that has OH group. This is the 4' that has the 5' carbon, CH2, and this one will bond to the phosphate group. This is the phosphate group, and this is the 5' carbon. This is the 3' carbon, and this is 1' that has the nitrogenous base such as adenine. This is a nucleotide. DNA polymerase can add new nucleotide and form bond between this is the phosphodiester bond between this 3' OH group and the 5' phosphate group of the next one. That means suppose this is another nucleotide. 5' carbon, this is on the 5' carbon, and it has oxygen, OH will bond with phosphate, and this will bond with this OA, O, and H will be removed. This is the phosphodiester bond. This is its 3' end, 3' OH. So the direction of synthesis of DNA is from 5' to 3'. Similarly, the antiparallel chain of the DNA will be 5' to 3'. Here it is 5', it is 3', and here it is 5', and it is 3'. Then they coil. So this is how DNA polymerase add new nucleotides. Scientists now have identified a total of 5 DNA polymerase. So DNA polymerase are of 5 types. That is 1, 2, 3, 4 and 5. This 1 and 3 is very important for DNA replication in E. coli, and they also act as a exonuclease. That means they can remove nucleotides, and this 2, 4 and 5 helps in repair. So DNA polymerase 3 is important, 1 and 3. The first successful DNA synthesis was done in a reaction mixture containing DNA fragments, number 2 dNTPs, and number 3 E. coli extract. So now what is dNTPs? The full form of dNTP is deoxyribonucleoside triphosphate. For example, DATP, DGTP. So what is deoxyribonucleoside? That is nucleoside with deoxyribose as its pentose sugar. For DATP, deoxyribose with adenine. This is deoxyribonucleoside and triphosphate. That means it has 3 phosphate group. These two bonds are called high energy bond. That means when they break, they release energy, and when they form, they require energy. So it is DATP. Just like that, it can be DGTP. You can comment it, what is DGTP. Collectively, they are called dNTPs. Scientists have found that 5 components are required for DNA synthesis. Number 1, this is all 4 dNTPs. Number 2 is DNA polymerase. Number 3 is E. coli DNA. Number 4 is primer. So what is primer? Primer is a small nucleotide that is synthesized by primase, and it will initiate DNA replication. So number 5 is magnesium for optimum activity of DNA polymerase. These 5 components are required for DNA replication. So the DNA polymerase can add new nucleotides. But how does DNA replication start? As we know that both the strands of a DNA is attached through hydrogen bond, and for DNA replication these two strands needs to be separate. Helicase is the enzyme, helicase, that breaks the bond between two strands of a DNA and also untwist them. This breaking is called denaturation of DNA or DNA melting. Suppose this is a untwisted DNA. Helicase acts here. So this type of structure is formed. Here is a helicase and here is a helicase. They move this direction. Helicase basically acts like a slider of a zipper. When they move, they open the two strands. The specific region where DNA denaturation occurs is called origin of replication. And this structure is called replication bubble. Replication bubble. This Y-shaped structure is called replication fork. Just like that, this is replication fork. So a replication bubble has two replication fork. The total structure is called replicon. Or replication unit. Cleared? So helicase acts like that. Now it is the time for second enzyme. Helicase is the first one and the second one is primase. Primase is an enzyme that forms a small RNA molecule. Basically, helicase recruits it. Suppose this is a replication fork. these are the template strand and helicase is here. Helicase recruits primase that will synthesize a small RNA nucleotide, and this RNA nucleotide will bind to this template strand. Suppose in this region, this is primer. So for this DNA, this is 5 prime, so it will be 3 prime, and it will be 3 prime, and it will be 5 prime. As this is the 3 prime, so for this primer it will be 5 prime and it will be 3 prime. Why do we need this RNA primer? Because DNA polymerase only can add nucleotides to its pre-existing one, but it cannot initiate the replication. So when this RNA primer is attached to this template, new nucleotide will bind on this position. So the new DNA strand will be synthesized on this direction, 5 prime to 3 prime direction. This is a continuous process. This strand is called leading strand. For this strand, this 3 prime to 5 prime strand, a new RNA primer is attached, suppose attached here in this region. So this is the 5 prime, so it will be the 3 prime and it will be the 5 prime position. As we know that DNA polymerase only can add a new nucleotide on 3 prime position, but this is 5 prime position. So DNA synthesis on this direction cannot be possible. So for this, a new primer, RNA primer will add here, suppose here. So it will be the 5 prime and it will be the 3 prime. Now new nucleotide can be added by DNA polymerase on this 3 prime position and in this direction. Again a primer can add here and DNA polymerase can add new nucleotide on this direction. So for this strand, it is not a continuous process, and this strand is called lagging strand. This is the discontinuous, and these fragments of DNA is called Okazaki fragment. So what is the difference between leading strand and lagging strand? For leading strand, it is DNA synthesis is a continuous process. For lagging strand, it is not a continuous process, it is discontinuous process. And for lagging strand, it needs many RNA primer, while the leading strand need only one RNA primer. So as the lagging strand has many RNA primer, this RNA primer needs to be cut. So the enzyme DNA polymerase 1, this will remove the RNA primer. Suppose they cut it. So now it is the 3 prime position. So DNA polymerase 3 will add new nucleotide on this and new DNA will be synthesized. But there will be a gap. Ligase is an enzyme that will fill the gap and seal it. So the fourth enzyme, the third enzyme is DNA polymerase 1 and 3, and the fourth enzyme is ligase that will seal the DNA fragments. So as the helicase move towards here, the bonds between the two DNA strands will break and this process will be continued. So a tension will be produced on this replication fork. So the fifth enzyme is Gyrase, which is actually a topoisomerase that will release the tension. So these are the enzymes, that is helicase, primase, polymerase, DNA polymerase, ligase and gyrase, that will help in DNA replication. This is the process of replication in prokaryotes. It is similar in eukaryotes, but there are some differences. As we know that prokaryotic cell has only single circular DNA in its cytoplasm, and the eukaryotic cell has chromosomes in their nucleus. So DNA replication in prokaryotes occurs in cytoplasm. But in eukaryotes, it happens in nucleus. And again, for circular DNA, there is only one replication bubble in prokaryotic cell. Just like, suppose this is a circular DNA and there is a replication bubble like this and this. In eukaryotes, chromosomes are linear and there are many replication bubbles. So just like that, this is a chromosome and there could be many replication bubbles like that. So in prokaryotes, single replication bubble is there. For eukaryotes, there are many replication bubbles. Another important difference is in prokaryotic replication, gyrase is needed, but in eukaryotic replication, gyrase is not needed. In prokaryotic replication, DNA polymerase 1 and 3 is involved. But for eukaryotic replication, DNA polymerase alpha, delta and epsilon is required. So what are their functions? DNA polymerase alpha will initiate the replication in eukaryotes, and delta is involved in synthesizing lagging strand and epsilon is involved in synthesizing leading strand. Another important difference in prokaryotic replication and in eukaryotic replication is in their okazaki fragment. In prokaryote, the okazaki fragments are long, about 1000 to 2000 nucleotide long. But in eukaryotes, the okazaki fragments are 100 to 200 nucleotides long. These are the 5 differences between prokaryotic and eukaryotic replication. I hope you understand this class. In our next class, we will discuss how DNA synthesize RNA.